Hi friends, if you like my videos, subscribe my channel and also press the bell icon for the latest updates. Thank you.
In this video, let us discuss about catabolism of amino acids. So before entering into this topic, you people should know what is meant by catabolism, right? Catabolism is nothing but the degradation of complex molecules to simple molecules is known as catabolism, or else the degradation of larger molecules to simpler molecules or small molecules is known as catabolism. And you have to apply this catabolism phenomena in amino acids. And based on the fate of carbon skeleton, I mean when you apply this catabolism phenomena amino acids, then these amino acids can be classified into two types: glycogenic amino acids and ketogenic amino acids. So what is meant by these glycogenic amino acids? The glycogenic amino acids can be defined as the glycogenic amino acids are defined as the glycogenic amino acids where the carbon skeleton will get degraded to alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, and pyruvate are known as glycogenic amino acids. So what are these alpha-ketoglutarate? What are all these? These are the intermediates of TCA cycle. You know about the TCA cycle, right? And this explanation I will say you here. So before going into explanation, let us learn the introduction part. And what is meant by ketogenic amino acids? The degradation of amino acids which forms the intermediates like acetyl-CoA or acetoacetate is known as ketogenic amino acids. So let us learn the examples of the glycogenic and ketogenic amino acids. Coming to the glycogenic amino acids examples: glycine, valine, methionine, threonine, histidine, asparagine, glutamine, alanine, cysteine, serine, arginine, proline, aspartate, and glutamine are the examples of glycogenic amino acids. So coming to the examples of ketogenic amino acids: leucine, tyrosine, tryptophan, phenylalanine, isoleucine are the examples of ketogenic amino acids. And the amino acids which exhibit the properties of both glycogenic and ketogenic are called glycoketogenic amino acids. And the examples of these amino acids are isoleucine, lysine, phenylalanine, tyrosine, and tryptophan. So these are the examples of glycogenic, ketogenic, and glycoketogenic amino acids. So I have said you that there will be integration in this TCA cycle, right? And normally there will form the intermediates. The amino acids which forms the intermediates are called amino acids. And if you see here, these are the TCA. This is the TCA cycle, and normally oxaloacetate will use acetyl-CoA and forms citrate. And from where this acetyl-CoA will be formed? From the pyruvate. And from where this pyruvate will be formed? Pyruvate will be formed from the glycolysis pathway, right? EMP pathway. I mean, this will be the end product of the glycolysis, and that pyruvate will form acetyl-CoA. With the help of this acetyl-CoA, oxaloacetate will form citrate, and this acetyl-CoA will also has a capacity to form acetoacetate. Okay, and now this citrate will form alpha-ketoglutarate, and here carbon dioxide will be liberated out, and the carbon dioxide which will be liberated out will be used further in the urea cycle, which I explained you before. So alpha-ketoglutarate will form succinyl-CoA, and this succinyl-CoA will form fumarate, and the fumarate will again form oxaloacetate in such a way that the oxaloacetate will again form the citrate by utilizing the acetyl-CoA which has been formed from the pyruvate. Okay, so in this method total cycle takes place, but here there will be no proper end product because this is a cycle process, so there will be no end product. So we will call each of them as intermediate compounds. So these are the intermediate compounds. So the amino acids which forms these intermediate compounds. So if you see here, normally I have given the examples of glycogenic amino acids, right? And if you see here, what I have given in the glycogenic amino acids, the degradation of amino acids which forms alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate. Alpha-ketoglutarate, if you see here, here the glycogenic amino acids will get degraded to form the alpha-ketoglutarate. That's what I given here. And succinyl-CoA, coming to the succinyl-CoA, here also the glycogenic amino acids will get degraded to form the succinyl-CoA. In that way, all of these intermediates which are present in this TCA cycle will be formed by the degradation of the glycogenic amino acids, but exception of two, that is acetyl-CoA and acetoacetate. Both of these acetyl-CoA and acetoacetate intermediates of TCA cycle will be formed by the ketogenic amino acids. And what are those amino acids I have given you here in the example? So now let us learn the degradation of the amino acids. I mean, I have said you that, for example, if you take alpha-ketoglutarate, I have said you that this is a glycogenic amino acid will get degraded to form the alpha-ketoglutarate, and let us see the phenomena how it gets degraded. Okay, so coming to the catabolism of glycogenic amino acids, glycine, alanine, serine, threonine, cysteine are the best examples. Normally there are many examples, but I cannot explain you all of this degradation metabolism, right? So I am going to explain you taking some of the examples only. So here I have took glycine, alanine, serine, threonine, and cysteine. So if you see here, the first substrate which acts in this reaction is alpha amino beta ketobutyrate. So this is the substrate, and this is the molecular structure of that alpha amino beta butyrate. and this will form threonine by utilizing an enzyme known as threonine dehydrogenase. And if you threonine hydrogenase, and if you see here in this threonine hydrogenase, here the hydrogenase is an enzyme which has been utilized over here. So what happens is that NADH plus and H plus will get reacted to form NAD plus. Okay, and here if you see here properly, here 2 H plus has been utilized, right? One of the H will be utilized here and another H will form another one here, like this. So totally the structure of threonine will be formed. So totally 2 H will be utilized by the NADH energy, and here one of the H will be joined here and another H will be joined towards another region of the carbon and forms threonine. And the enzyme which has been utilized here is threonine hydrogenase. Don't forget to write the enzymes. So next, this threonine, so here if you see here the threonine formation has been completed, right? And next let us see the formation of glycine. So normally this threonine will form glycine by utilizing an enzyme known as serine hydroxymethyl transferase. And if you see here, normally this threonine will get degraded and forms acetaldehyde. So if you see here how the acetaldehyde will be formed, CH3, C double bond O, and only one H will be taken. So normally this part will be degraded out. Let us see here. Up to here the part will get degraded out as acetaldehyde, and next the remaining part will form glycine. Okay, if you see here properly, H, CH, NH3, CO has been left over there, right? And that will form the glycine. Okay, by utilizing an enzyme known as serine hydroxymethyl transferase. And here acetaldehyde has been formed, right? And if you see here alpha amino beta ketobutyrate, by utilizing an enzyme known as alpha amino ketobutyrate lyase, it will form acetyl-CoA, right? It will form the acetyl-CoA, because if you take here COSH has been introduced into it, where the H part will get removed, and instead of that HA, what happens the COH which has been added will get binded over with this acetaldehyde. Okay, and aldehyde group will be removed out. It's simple to say, and forms acetyl-CoA because COSH group has been introduced into it. And this reaction takes place by introducing an enzyme known as alpha amino beta ketobutyrate lyase, and it forms glycine, right? And if you see here this glycine, and here if you see normally we have completed about the degradation of threonine as well as the glycine. So now let us see another remaining 3 amino acids. So from the glycine it forms serine by utilizing an enzyme known as serine hydroxymethyl transferase. And if you see here methyl transferase is nothing but transamination process takes place between this reaction in such a way that the methyl group will get transferred over here. Okay, CH2 is a methyl group which will get transferred over here. And how the methyl group will get transferred and what is meant by the transamination process, I already explained in my previous videos, and the link of that video will be given in the description box, so people who are interested, you can watch that video. And next, the alanine will form normally the serine which has been formed here will form pyruvate by serine dehydratase. This will be formed. So here again what happens is that the transamination process takes place in such a way that the keto group will get converted to amino group. So what is meant by transamination? What is meant by transamination is that the transfer of amino group to keto group in such a way that the amino acid should convert into keto acid is known as transamination process. So if you see here, this keto group will get this amino group will get converted to keto group, right? Because of the transamination process, and the enzyme which has been utilized over here is known as serine dehydratase. Okay, and next if you see here alanine, this alanine will again get converted into pyruvate. And here if you see here, this is again the transamination process takes place here, where the amino group will get transferred in such a way that to form the pyruvate, to form keto acid, in simple words to say here. And here alpha-ketoglutarate and glutamate will get alpha-ketoglutarate will be utilized and the glutamate will be released out. And next, this is the structure of the cysteine. And remember here the cysteine consists of sulfur group in its structure, and that cysteine with the help of a enzyme known as cysteine desulfidase, what happens is that this sulfur group will get removed out in such a way that it forms pyruvate. Okay, so normally here what happens, normally we have degraded glycine, alanine, serine, threonine, cysteine. So here threonine, next alanine, here we have formed alanine, and next glycine, and next cysteine, and next threonine. So like this serine, so like this we have formed the catabolism of glycogenic amino acids. So now let us see catabolism of some other amino acids. So if you see here the catabolism of, I am going to explain you about the catabolism of arginine, proline, histidine, glutamine, glutamate. So if you see here, this is the structure of histidine, and with the help of an enzyme known as histidine ammonia lyase, where the ammonia group will get liberated out and it forms urosinate, and with the help of an enzyme known as urosinate hydratase, where the hydrogen molecule will be utilized by water molecule, which will get introduced during this reaction and forms 4-imidazole 5-propionate. And now here another water molecule will be utilized here, and with the help of an enzyme known as propionase, it forms N-formamino glutamate, right? And with the help of an enzyme known as imidazolone formamine transferase, then what happens is that it forms glutamate. Several reactions takes place here. So with the help of the several reactions, the final product which will be formed is glutamate, and from the glutamate it forms alpha-ketoglutarate. And if you see here, the tetrahydrofluoride will be utilized over here, and what will be formed here is glutamate, and from the glutamate alpha-ketoglutarate will be formed. So if you see here, alpha-ketoglutarate will be formed, right? So if you see in my previous case, in the integration of TCA cycle, this alpha-ketoglutarate will be formed by the histidine, right? So here in this structure also I explained you about histidine. So histidine undergoes degradation process like this and finally forms alpha-ketoglutarate, right? Next, let us take another amino acid. Let us take now proline. So we have completed about histidine. Let us take about the proline, and this will be the molecular structure of the proline. And this proline undergoes oxidation reaction by utilizing an enzyme known as proline oxidase, and here oxygen, half of the molecule of oxygen will be utilized here and releases out as a water molecule and forms proline 5-carboxylase, and it undergoes several reactions. I didn't mention here enzyme because many reactions takes place here. I can't explain many reactions because it takes up to 3 to 4 pages, and those many reactions it undergoes, I can't explain you that. So just keep remember like this. It undergoes many reactions, so I didn't mention any enzyme here, and when it undergoes many reactions, then it forms glutamate 5-semialdehyde. And now the glutamate 5-semialdehyde will further form glutamate, and from the glutamate it forms alpha-ketoglutarate, right? So what is the enzyme? So what is the amino acid which has been used here is proline, right? And if you see in the previous case like this, here what is alpha-ketoglutarate formation will be takes place by the proline. So we have completed about histidine as well as the proline. So now let us see in the case of arginine. So this is the structure of the arginine. So here this arginine plays a major role in urea cycle, and I have explained you about urea cycle in my previous videos, and the link will be given in the description box, students. And next, here urea will be liberated out when the conversion of arginine into ornithine takes place, and during this conversion of urea, one of the water molecule will be utilized. With the help of that water molecule only, the urea will be exhibited out. And here the enzyme which plays a major role in the conversion of arginine to ornithine is arginase. And now ornithine, which has been formed over here, will get transferred into, or else will get converted into glutamate 5-semialdehyde by utilizing an enzyme known as amino transferase. And if you see here, what happens is that semialdehyde, how you can identify the semialdehyde, because O here O and here H group, and here normally this group will get removed in such a way that due to the process of transamination. What is meant by transamination? I already explained you. The conversion of, or as the transfer of amino group to the keto group in such a way that the amino acid will form the keto acid is known as transamination process, right? So here this amino group will get transferred to form the keto group, and this will form the glutamate 5-semialdehyde by utilizing an enzyme known as amino transferase, and here alpha-ketoglutarate will be utilized and the glutamate will be released out. And this phenomenon you can understand when only you can know about the transamination process. So what I wanted to suggest you friends is that before watching this video, you should watch this transamination process, deamination process, and urea cycle, carboxylation. If you watch those videos, then only you can understand this video properly. Okay. And next, because it is a complex, it is a very complex theory to understand, so that's only the reason I am recommending you to watch that videos, and all of that videos link will be given in the description box. Okay. And now this glutamate 5-semialdehyde will undergoes dehydrogenation process with the help of an enzyme known as dehydrogenase, and here NADP energy will be used and NADPHP energy will be liberated out, and it forms glutamate, and this glutamate will further forms alpha-ketoglutarate, and the enzyme which has been utilized over here is glutamate dehydrogenase. Of course, all of them will form glutamate, and this acts as intermediate compound, and the final end product which will be formed is alpha-ketoglutarate. Here only the end product of the alpha-ketoglutarate will be formed, but if you take in the case of TCA cycle, this alpha-ketoglutarate will not form as the end product because the cycle gets continuous, so this acts as an intermediate compound only, but only in this catabolism process this alpha-ketoglutarate acts as the end product. Next, glutamine. Here glutamine, this will be the molecular structure of glutamine. How you can say this is a glutamine? Because NH2 group will be present over here. And now amine glutamine. Okay, this glutamine will undergoes with the help of an enzyme known as glutaminase. This will form the glutamate. So if you see here, COO group will be attached to it. Okay, with the help of an enzyme known as glutaminase, and here water molecule will be used and the amino group will be removed out. So by this, what did you understand? Here the deamination process takes place because the removal of amino group from the structure. If you see here, the amino group has been liberated out, right? So when you introduce water molecule, what happens is that here the normally, wait a second, this will be the molecular structure of water molecule, right? So water molecule consists of oxygen group, and this amino group will be liberated out, and this oxygen group will get bonded over with keto group in such a way that it forms carboxyl group, COO minus. Okay, and thus forms glutamate. So in this way, we have learnt about arginine, proline, histidine, and glutamine. In the case of glutamate, if you see here, this glutamate will get converted into alpha-ketoglutarate by utilizing an enzyme known as glutamate dehydrogenase. So if you see here, deamination process has been taken place here. So how this deamination process takes place, I have already explained in my previous video. I will give the link in the description box. You can watch it. And here the NP plus energy will be required here, and NADPH and NS3, ammonia will get liberated out. But because of the process called deamination, ammonia will get liberated out. So in this way, we have learnt about arginine, proline, okay, and histidine, and next glutamine and glutamate. Here glutamine, we have learnt about the glutamate dehydrogenase, glutamine, and as well as the glutamate. So in this way, the catabolism of this amino acids takes place. Okay, in other words to say, degradation of the amino acids like this. So now let us learn about the degradation of phenylalanine and tyrosine, or as the catabolism of phenylalanine or tyrosine. So if you see here, this is the molecular structure of phenylalanine, and this phenylalanine will form tyrosine, and the enzyme which has been utilized over here is known as phenylalanine hydroxylase. Okay, and here tetrahydrobiopterin along with the oxygen will be utilized over here, and the dihydrobiopterin along with the water molecule will get released out. Okay, and this will be the molecular structure of the tyrosine, as we required here oxygen molecule and the H2O molecule, because it forms the OH group here, right? OH group will be formed here. And if you see here, with the help of an again transaminase enzyme, and this undergoes transamination reaction in such a way that the amino group will get liberated out. This amino group will get transferred into keto group in such a way that this amino acid will get converted into keto group. So here this is a keto group, and this amino group will get shifted into the keto group in such a way that this amino acid will get converted into keto acid. So it is called as para-hydroxyphenyl pyruvate, and here alpha-ketoglutarate will be utilized in such a way that to form glutamate. And next, now here, with the help of an enzyme known as para-hydroxyphenyl pyruvate dehydrogenase, here dioxidase is nothing but oxygen will be utilized over here, and a water molecule along with the carbon dioxide will get liberated out in such a way that it forms homogentisate. Okay, and now this homogentisate, which has been formed with the help of an enzyme known as homogentisate dioxidase, will form again 4-methyl acetoacetate. So if you see here, normally what happens in this reaction is that nothing but cleavage of the bond takes place over here. So if you see here, cleavage of the bond takes place. Due to this cleavage of the bonds, then what happened is that the total cycle, this benzene cycle will get disrupted in such a way that it forms the structure known as 4-methyl acetoacetate. And with the help of an enzyme known as isomerase, this will form 4-fumarate acetoacetate. Okay, and if you see here properly, what is meant by, if you use isomerase enzyme, then nothing but the carbon position will get shifted out. This carboxyl group which has been attached to this here, this has been represented towards right side, right, and it will be represented towards left side if you use an enzyme known as isomerase. And next, by utilizing an enzyme known as isomerase, it forms 4-fumarate acetoacetate, I have said you right. And now here again water molecule will be utilized over here. So water molecule, this will be the molecular structure of the water, right? And if you see here, wait a second, cleavage of the bond takes place over here, right? And this will be the molecular structure of water molecule, and if you see in the structure, it forms fumarate and the acetoacetate, because the cleavage of the bond takes place by the one of the hydrogen which is present in the water molecule. And if you see here, one of the hydrogen helps in the cleavage of the structure, and another one O oxygen which has been present over here will form CO over here, and another H which has been represented over here will form the CH3. One of the hydrogen which has been present over here will get attached to this to this group and form CH3. So the one of the hydrogen will form the cleavage, and another hydrogen will get combined with this CH2 and form CH3, and another oxygen will get combined with this CO and forms carboxyl group, in such a way that as it undergoes cleavage, this will get separated and this part will get separated, and this part is said to be as fumarate and the another part is said to be as acetate. So this is about the degradation of the amino acids, and thank you for watching this video, guys. Hope you guys will understand this topic as much as possible, and I tried my best. Thank you for watching this video, guys. If you like this video, please do like and subscribe, and if you have any doubts regarding this video, please comment in the comment box. Thank you.